Okay, so hello and welcome to today's live class session of week 12. So this is the last week of session that will be held for this year's course on experimental biochemistry. And myself, I am Srijan Sen from IIT Kharagpur, and today I will be taking the week 12 question answer session where I would be discussing the lectures in brief and then followed by solving a few questions that will help you in answering in the upcoming exam, probably, which is going to be held in the next week, or the actual date I'm not aware of. But anyway, so this lecture will definitely help you even for the competitive exams that you would be giving in the upcoming days or in the near future. So yeah, good evening. So okay, so I'm sharing my slide with you all. Kindly confirm. Yeah, so I guess my screen is visible to you. Okay, so let us start with the slide. Fantastic. Okay, do you have any questions from the previous class or previous classes? Any questions from the previous lectures? No questions. So okay, so let us start with today's one. Great. So in today's lecture, we would be mostly discussing about blotting, where we'd be discussing about the major types of blotting techniques, broadly classifying them under immunoassay techniques. So mostly today's class will deal with blotting and what are the different applications of the different blotting and all. So we would be talking about that. So before we directly jump into blotting, first let us understand what blotting is. So blotting is a technique that is used for transferring DNA, RNA, different nucleic acids, proteins onto a carrier such that they can be separated, and often follows the use of a gel electrophoresis. Like the southern blot is used for transferring of DNA, the northern blot for RNA, and western blot for proteins. So basically these are the three major blotting techniques that we would be studying today. Like we have so these are the different blotting techniques. Like we have a geographical compass. This is also represents a biological compass where we have the different like the different directions representing the different types of blots. Starting from the northern blot, we have we use it for the northern blot, we use it for detecting of RNA. Moving to the southern blot, it's used for the detecting of DNA. Western blot for detecting proteins, and eastern blot for detecting protein post-translational modifications, or PTMs. Okay, so these are the four different types of blotting techniques that are largely used. Apart from that, there are some subdivisions, like you have the northeast blotting, you have the east-east blotting, you have the southeast blotting, the southwest blotting, etc. And there are a lot of other blotting techniques, but majorly these are the four blotting techniques that we use. and three of them being the major ones like northern, southern, and western blot. So starting with a very interesting blotting technique, which is known as the southern blotting. It was named after E.M. Southern in 1975. So the other names, so the first blotting technique that was developed for testing on DNA was southern blotting by E.M. Southern. The other blotting techniques were not named after scientists. So basically when you write them, it's written southern with the like you have caps on, like you write in the caps letter, okay, southern. But for the other blotting techniques, you do not use the caps, you write them in small. Okay, so like for northern or western or any other blotting techniques. So this was the only blotting technique that was named after a scientist, and the other names came based on this. So southern, northern, eastern, and western. That sounds funny. Okay. The southern blot is used to detect the presence of a particular piece of DNA in a sample. So we know that southern blot is basically used for detecting of DNA in a sample, and it can be basically a single gene or can be part of a larger piece of DNA, like you have a viral genome. So basically what you do in southern blotting is it combines agarose gel electrophoresis for separation. So you separate the DNAs from the other mixtures using an agarose gel electrophoresis, and you transfer the size separated DNA to a filter membrane for the probe hybridization. So this technique basically includes a hybridization technique, or known as the probe hybridization. So the key to this method is hybridization. So what is hybridization? It is a process of forming double-stranded DNA molecule between a single-stranded DNA probe and a single-stranded target patient DNA. So how do we know that where this probe is going to go and bind? So we know what is the target sequence of the DNA that we are wanting to find. So the probe would be designed in such a manner. This probe can be fluorescent level, this probe may not be fluorescent level. That is not our concern. But what is our concern is that the sequence, or that is the genome sequence, is basically known to us. It can be the genome sequence, it can be the small DNA sequence, but that should be known to us, and the probe should have a sequence that is complementary, exactly complementary to that particular DNA sequence. Now this probe will go and bind, so this particular probe will go and bind to only that portion of the DNA to which it is complementary, and not go and bind to non-specific region. So that is why we call this hybridization, and this is very important for forming the double-stranded DNA. And this double-stranded DNA, if that probe is fluorescently labeled, it will glow under the fluorescence channel. So the principle behind this southern blotting was that the mixture of molecules are separated and immobilized on a matrix. So the probe is added to the matrix to bind to the specific DNA into the specific region, and then you wash the particular membrane, that is the matrix, to remove any unbound probes or any unbound molecules, and the place the probe is connected corresponds to the location of the immobilized target molecule. So this is basically the apparatus. You have a gel running apparatus where you separate them according to the size, and then this is the transfer medium. This is the transfer apparatus where you transfer the DNA onto a particular matrix. That can be your polyvinyl, polyvinyl chloride matrix, polyvinyl fluoride matrix, or even there are other nitrocellulose membrane that you can also basically use. So what you do over here for the transfer, you have, so basically we start from the bottom where you have a plastic tray, and there is an electrode system that will be used for flowing of the current. And over here you have the gel, you see you have a plastic tray. Over on this you would be transferring it. You have the two electrodes attached over here, and then you place the gel, you place the membrane, you provide on that the filter paper, blotting paper, you have the blotting paper, some paper tissues on a glass plate and a weight such that it keeps them, the weight is maintained, and then you transfer the current and the gel will start transferring the DNA materials, all the materials onto this particular membrane via the passing of electricity, a particular current. So basically for blotting we use very low voltage of current, not like we use for a gel running for gel electrophoresis, and also minimalistic current is used. So what do you basically do in southern blotting? So we digest the DNA with an appropriate restriction enzyme. So the complex mixture of fragments is subjected to gel electrophoresis to separate the fragments according to their size. So you get different sizes of molecules over here and you observe them. Now what happens, you can see over here that there is an electricity that is passing because the salt solution, so this is a buffer solution, so you have the ions over here that would maintain the you can say the electricity, okay, the current that is flowing. So the restriction fragments present in the gel are denatured with alkali and transferred onto a nitrocellulose filter or a nylon membrane by blotting. So you blot them and transfer into the different nitrocellulose membrane or the nylon membrane. And this procedure basically preserves the distribution of fragments in the gel, creating a replica of the gel on the filter. So how you transfer it depends on the replica that is created on the membrane, and this will help you to identify the molecules. Now what you do, now all the nucleic acids, basically all the DNA molecules are transferred onto the membrane. Now you just discard the gel because it is of no use. What you do is you take the membrane, because now the membrane is very useful to you. So the membrane or the filter is incubated under hybridization conditions with radio labeled DNA probe. So the probe is radio labeled, and then what you do, the probe hybridizes to the complementary DNA restriction fragment. It goes and binds over there, like you see. It will go and bind only to A but not to B because it is not complementary in that manner. And then you wash away the excess probe, the excess material, and the probe is bound to the filter, is detected by autoradiography, which reveals the DNA fragment to which the probe is hybridized. So from there you can tell that which portion of your DNA is actually containing your desired product. So summarizing the entire procedure, what we do, first we extract and purify the DNA. Then it is restriction digested with different enzymes and separated by electrophoresis. Then what you do, you alkaline denature the DNA, you transfer it to a nitrocellulose membrane and block with the excess DNA. You hybridize it with a probe, and then you wash away the unbound probe and then develop the blot using autoradiography. So this is just a table for this is just a poster form where you can see all the steps over here. First digestion, then subjecting it to agarose electrophoresis for separation, then transfer. So there are different materials. You have a stack of paper towels, you have nitrocellulose membrane, the gel is at the bottom, you have the alkali solution, and then comes the incubation step for hybridization using radio labeled DNA probe. Excuse me. and the probe hybridizes to the complementary DNA restriction fragment. You wash away the excess probe and observe under the autoradiograph. So the different applications of southern blotting includes your gene discovery, evolution and developmental studies. Then you have mapping, diagnostics, forensics. Also it is used for DNA fingerprinting and RFLP maps. Also you can use this for the identification of transferred genes in transgenic individuals. So now the question is why do we even use blotting? We can directly see it on a gel. Yes, but there are a mixture of different fragments. Okay, so there can be several fragments of the same size, of the same length. Okay, but varies in the sequence. So for that to identify the exact sequence where our exact desired product is, we need to perform southern blotting. Okay, or any blotting to exactly or to appropriately tell where our desired product lies. Now, in a similar manner, this northern blotting was also developed, but we performed this for RNA. So it is a technique for detection of specific RNA sequences. It was developed by James Alvin, not someone, not Dr. Northern or Mr. Northern and all. It was developed by Alvin and Stark at Stanford University, 1979, and was named as by analogy to southern blotting. So what are the steps basically involved? So you isolate RNA from several samples like tissues and some developmental stages of the same tissue, and we know that the RNA is more susceptible to degradation than DNA, so we have to work more cautiously. So the sample is loaded on gel and the RNA samples are separated according to their size on the agarose gel. So the resulting gel following is the electrophoresis run, where you separate all the RNA molecules, and then it is blotted on a nylon membrane in a similar manner like we do for southern blotting, where you have the paper towels on the top, you have the membrane, then you have the gel, and finally the sponge, or you can even have the filter papers, and all followed by the salt solution that will be useful for transfer of electricity in a proper manner. So now when you do it, so now you blot it on a nylon membrane or nitrocellulose filter paper, even benzyl oxymethyl papers can be used for the transfer. So this is the filter. You have the gel over here that is entirely transferred onto the filter. Then it is placed on a dish containing the hybridization buffer with the labeled probe. The similar kinds of probes are used for the hybridization process. So the probe will hybridize to the RNA on the blot that corresponds to the sequence of interest, and the membrane is washed to remove the unbound probe. And then via using autoradiogram or an X-ray film, you can do autoradiography or chemiluminescence reaction if it is chemically labeled probe. So in both the cases you see a dark band on an X-ray film, and now the expression patterns of the sequence of interest can be compared. So first you isolate from biological samples, then you run it on a gel to separate them according to their size, and you use 32p labeled size markers. It is transferred onto a gel. It is transferred onto a membrane that can be a nylon membrane or an amino benzoyl oxymethyl filter paper. Then you hybridize it using a particular probe having the complementary sequence, and finally you observe it by autoradiography or chemiluminescence. And in both the cases you observe a dark band. From there you can tell which is your desired RNA sequence or your desired product. So the several applications of northern blotting includes the standard for the study of gene expression at an mRNA level. You can detect the mRNA transcript size, study the RNA degradation, study RNA splicing, study RNA half-life, and it is often used to confirm and check transgenic knockout mice animals. So the disadvantages of northern blotting is that, first of all, you're working with an RNA, so it is very susceptible to degradation. It gets easily degraded. So if RNA samples are slightly degraded by RNases, the quality of the data and the quantification of the expression is negatively affected. Now, the standard of the northern blot method is slightly less sensitive, and detection with multiple probes becomes a major problem. Now coming to proteins, Western blotting that deals with proteins for the detection of proteins. So Western blotting is an immunoblotting technique that relies upon the specificity of binding between a protein of interest and a probe that is basically an antibody raised against a particular protein. Like you have an histag antibody against that is grown in mouse. So his mouse antibody for the detection of proteins of interest in a mixture of many other similar molecules. So like we used agarose gel electrophoresis for separation of DNA and RNA. Over here we use an SDS page technique for Western blotting for the separation of molecules. So what we do, a protein sample is subjected to electrophoresis on an SDS page, and electroblotting transfers the separated proteins from the gel to a nitrocellulose membrane. Again, polyvinyl difluoride membranes can be used. Now the blot is incubated with generic proteins such as milk or BSA that binds to the remaining sticky places on the nitrocellulose. It is used as a blocking agent. So an antibody that is specific for the protein of interest is added to the nitrocellulose sheet and reacts with the antigen, and this is kept overnight at cold temperature. So only the band containing the protein of interest binds to the antibody, forming a layer of the antibody molecules. The next day you wash it to remove any unbound antibodies, and then you add the secondary antibody in a similar solution of BSA or milk, and then you keep it in rocking condition for about an hour or so, and you go for autoradiography. So what you do after washing, for the removal of non-specifically bound antibody 1, the secondary antibody is added, which specifically recognizes the FC region of the primary antibody and binds it. Now, the secondary antibody is radioactively labeled or covalently linked to a reporter enzyme that allows to visualize the protein antibody 1, antibody 2 complex. So this is the entire protocol where you prepare the samples first, you extract the plasmid, you grow them, you grow all the proteins, you run it, so this is the protein extraction. Then you boil it, sonicate it, homogenize it. Even if you want to purify it, you can purify it and then run it on an SDS page to get the different, you can see the different protein bands over here. And then for the protein transfer, you have the filter pads, you have the membrane, the gel, and you have another set of filter pads, and then you put it, you stack it and you put it, and you let the transfer to happen. So after the transfer, you can see from the SDS gel it has moved on to the membrane, and this membrane is kept for first for blocking by BSA and or you can say milk proteins. Then you use antibody incubation, first with the primary antibody, then with the secondary antibody, and finally you develop the blot using luminescence detection or fluorescence detection or even extra film development. So the similar protocol is also written over here, which you can follow for performing the blotting techniques. The applications of this blotting is used basically for the confirmatory HIV test. Western blot is used as a definitive test for bovine spongiform encephalopathy, and some of the Lyme diseases testing employ Western blotting. So these are the different applications of these three techniques, and these are the important techniques that you need to keep in mind whenever you are studying about this particular lecture. So if you have any questions on up till now about this particular lecture, you can definitely ask me. This is going to be a very short lecture. Okay, so you had a question in assignment 11, question number 9. Okay, for that I need to get back to that. So let me see if I have those questions. Just give me a minute. I will just get back to you with the question. Yeah, tell me, what is your regarding KD when K is given in the presence of a competitive inhibitor? Yeah, so the question that it says is that you are given an association constant, like you have Ka, that is 10 to the power 7 molar inverse, right? And what is most likely to be the measured Kd in the presence of a competing inhibitor? So you are given Ka and you have to find the Kd. So what we know, lower the Kd value, better is the binding. Higher the Kd value, poor is the binding, or the binding becomes poor. So now in a normal case scenario, what happens? You have Ka and now you have to find Kd. So Ka is basically 1, it is the reciprocal of Kd. So 1 by Kd equals to Ka. So in a normal case scenario, what would have happened? It is 10 to the power minus 7 molar. That would have been Kd. Fine. Now there is a competitive inhibitor. So what is a competitive inhibitor doing? So you have a protein that has a particular binding site, or you can say an active site where the molecule goes and binds. Now what happens, there is another very interesting inhibitor that also has a similar kind of binding to that particular position or that particular site. So what happens, in one site there are two species that are wanting to bind, and so there are two species that is wanting to bind. So now you imagine, if you do not want, so the protein does not know who we want to bind, because the protein is neither our friend nor can it hear us. So if even if we want a particular ligand to go and bind, like ligand X is the one that we want our protein, that we want the protein to bind to, but there is another uh, you can say another ligand Y that is acting as a competitive inhibitor for the binding of X to the particular protein. Now what happens, the protein does not know, because the protein does not know that it has to only catch X. It can catch X, it can catch Y, it can catch even other molecules if it has good binding affinity. Now what happens in this case, there is an equilibrium that is being maintained. Like you have the protein, you have X and Y. So the moment X is binding, it also has a tendency to, the Y also has a tendency to go and bind to that particular position. So X just moves out, Y binds. Y moves out, X binds. X moves out, Y binds. And this is a cycle that is basically going on. So there is an equilibrium. In no case only the X will bind or only the Y will bind. Okay. Now, so that place can be occupied either by X or by Y. But what happens? So only the protein binding to X has an association constant of 10 to the power 7 or a dissociation constant of 10 to the power minus 7. Now, in the normal scenario, it would have occurred is 10 to the power minus 7. But the moment there is a competing inhibitor or the place can be occupied by more than one species, the association constant decreases. So if you see the options, so by how much it decreases, it is very difficult to say unless we know the binding affinities of the individual molecules. We only know for X, but we do not know for the competing inhibitor. So what we can tell is that the association constant will decrease. So from 10 to the power 7, it decreases down to say 10 to the power 6. So it decreases by 10-fold. Now when you take the Kd in the presence of a competing inhibitor, it becomes 10 to the power minus 6. So the options that you can see is 10 to the power minus 6, 10 to the power 6, that is incorrect, and 10 to the power minus 7 and 10 to the power minus 8. So 10 to the power minus 8 means that is having a stronger affinity. But here the affinity is getting weaker because there is another species that is also wanting to bind. So it will not remain the same, it will not increase, rather it will decrease. So it becomes 10 to the power minus 6. Okay?
Susan, but what they have given in the paper is that the assignment is that they have mentioned Ka as 10 power 16. Okay, am I audible to you?
Yes, yes. So are you
Uh,
talking about the assignment 11
yeah,
of,
assignment. Yeah, assignment 11,
oh,
question
basically you
number
do not
9.
have the question of that particular assignment. It would be great if you can just email me that, then I can get back
Yeah,
with an
but what
answer.
they have given is I followed your last class. I understood well, but what they have given is 10 power minus 16 KD. They have given it as 10 power minus 16 in the presence of a competitive inhibitor. So that's my query.
Okay, but I need to check the question once. It would
Okay,
you
okay.
just email me once. I
And
will.
in the options also they have given one is 10 power minus 10, the other one is 10 power minus 8.
Okay, so the question is
Very
similar type,
near.
but they have twisted the
Yeah.
question. Okay, so I
Yeah.
will
Oh,
check
yeah.
that. I will check once and I will give you the answer. Okay.
Okay. Okay. Okay, Srijan.
Okay.
Okay. Thank you.
Okay. Any other doubts you have? Okay, even if you're having any
No, no, no.
doubts.
Continue with this. Continue with the assignment
Okay,
of
fine.
this week. Okay.
Okay, fine. Right. So, okay, we will do with the questions then. Yeah. So this one. So the first question. So it says, what is the first step of Western blot? So from here, basically, you can expect direct questions. Nothing much to. It won't be very difficult for you to answer. These are basically direct questions,
Yeah.
so you can.
Yeah. Run a STS page.
Yeah, that's correct. So to run an SDS page, that is the first step that you do for a Western blot. So Western blotting is performed to detect.
Protein. Specific protein.
Also correct. Next one, Western blotting technique is also known as.
Immunoblotting.
The proteins in a sample are separated using
SDS page.
correct. For DNA fingerprinting, which of the following technique is used? Southern blotting, northern
Southern
blotting.
block. Southern blotting.
Is also correct answer. So you can see these are the questions. These are very direct.
Very
Very
simple questions. Yeah.
yeah. So okay, the next one. In blotting, sorry. In blotting technique, a probe is a specific protein to detect DNA, a short piece of labeled double-stranded DNA complemented with a specific DNA. It's a short piece of single-stranded DNA or RNA complementary to the specific DNA or RNA and the specific protein to detect RNA.
It is a C.
See?
A short piece of labeled single standard DNA.
Exactly, right. Very good. So it is C. That is correct. The next one. Which of the technique is suitable for detecting a specific mRNA expression in cell?
Northern blotting.
Okay. The non-specific sites on the PVDF membrane can be blocked by. So for blocking the non-specific sites, what do you use for a PVDF membrane?
We use BSA.
Yes, we definitely use BSA. Apart from that, we can also use non-fat dry milk or skimmed milk, which we also say skimmed milk powder, which we dissolve in a particular buffer and then we use it as a blocking agent. Why the probe is labeled? To improve binding capability, to improve location
Location
identification.
identification.
Yes. To improve the location identification. That is correct answer. The next one. What is the major force behind protein and nucleic acids to go out of the gel onto the membrane? Electric current, pH gradient, voltage, temperature.
Electric current.
Yes. Any other option? Any other option you feel is correct?
pH gradient.
No, basically the pH is not an issue, so it's very directly related. If you have an electric current, you also got to have the voltage. So basically,
Yeah.
for Western blotting, we use for the transfer 20 volts and 150 milliamperes. Excuse me. Okay. Why labeled antibodies are used? Why we have used labeled antibodies?
To detect the presence of a particular protein molecule in Western blotting.
Yes, very good. That's the answer. So C is the answer. Which of the following is or are used to detect the protein in Western blotting? It's also easy. Antigen, antibody, DNA probe, RNA probe. So what do you use to detect protein in Western blotting?
Antibodies.
Which of the following membrane is best to use if you want to strip an already used membrane for Western blotting?
It is a PVDF membrane.
Right. Fantastic. It's the PVDF membrane that you use if you want to reuse the membrane. First, if you have used for Western blotting, now you want to reuse the membrane, you can use the PVDF. If you forget washing the membrane after incubation with primary secondary antibody, what will happen?
So I think many options are correct here.
Yeah, so
Uh, yeah, A is one of the options, and
sorry, I did not get you. First one, A,
A,
yes, background noise will be high.
so it will be high. Uh, whole blot will be dark. No specific band will be observed.
Fantastic. So all the three options are correct over here.
Yeah,
And the last one, why ghost bands appear in blots?
maybe too much protein has been loaded.
Yeah. Any other?
I think that's the correct one.
The other option that is correct is
Last maybe.
incubation.
Incubation. Yeah,
Maybe
yeah,
the
correct.
primary antibody was
For a
stored.
too long time. Yeah, yeah, makes sense. Yeah.
So these are the two options that are basically correct. Okay. So
Yes,
that
yes,
is
yes.
what
Yeah.
I have from my portion. Okay. So if there are no doubts, then we can say give your exams nicely. So when do you have when do you have the exam?
20th, next Saturday is the exam. Thank you very much for. I have gone through many of your videos. They're very useful. You explained
Okay.
very nicely. Thank you very much.
Okay. Best of luck for your exam. Give it nicely.
Thank you.
Just
Thank
practice
you.
the
Thank
live
you.
sessions. You can do
Yeah.
good. Okay.
Yeah. Thank you. Thank you,
Thank you
Sijan.
so
Bye.
much. So best of luck to you all for your upcoming exam. I hope that you can do a great job in the exam and just go through the lectures very nicely. Just go through whatever has been discussed in the live session. Okay, so whatever has been discussed in the live session will be very useful to you all. So go through them, and I guess you can do good in the exam. Okay, and apart from that, if you have any questions regarding anything you want to ask at any point of time, you can get back to me in mail. I'm attaching my mail ID over here, so you can definitely get back to me anytime. So it's not a big deal, and I would be very happy to help you out. Okay, so have a great time, and these lectures are anyways recorded, so if at any point of time you feel you are facing any difficulty, do get back to look at the lectures in YouTube, and I think that would definitely help you, right? So once again, best of luck for your exam, and so this is basically the last class. I hope you all the best for your future, and hope you would all be doing great job in your part of life. Okay, so thank you all. Thank you very much for attending the classes, and that's all. Okay.